What you are looking at is the most sophisticated pump ever engineered, and it was built entirely from scratch inside a developing embryo, without a blueprint, without external tools, and without ever stopping to rest. The human heart begins as a microscopic cluster of mesodermal cells and transforms, over a matter of weeks, into a four-chambered, pressure-regulated, electrically synchronized organ capable of beating over 3 billion times across a human lifetime. This video traces that entire construction journey, from the very first blood islands forming in the yolk sac through the folding of a primitive horseshoe-shaped tissue mass, through the extraordinary looping of a beating straight tube, all the way to the final pressure-driven seal that occurs at the precise moment of birth. Every step is clinically essential. Every step has a corresponding congenital defect when it goes wrong. This is cardiovascular embryology, and understanding it is non-negotiable for MBBS, USMLE Step 1, and clinical medicine.
Before we dive deep, here is the master framework you need locked into your memory. The entire story of cardiovascular development unfolds across 4 sequential phases. Phase 1 is hemopoiesis, the simultaneous origin of blood cells and the blood vessels that will carry them, driven by a remarkable hemodynamic principle. Phase 2 is the origin of the early heart, how a horseshoe-shaped slab of mesoderm folds, fuses at the midline, and becomes the primitive beating tube. Phase 3 is cardiac looping, one of the most spatially complex events in all of embryology, where a straight 2-chambered tube must fold itself into the 3-dimensional architecture that positions each future chamber in its correct anatomical address. And Phase 4 is septation, the simultaneous construction of internal walls across all 4 chambers while the heart continues pumping blood without interruption. Each phase builds irreversibly on the last. Miss the sequence and the defects become impossible to understand.
Before a single cardiac cell contracts, the embryo must solve an urgent problem. It is growing too fast for passive diffusion to deliver oxygen to its cells. The solution begins in the yolk sac, where clusters of mesodermal precursor cells called hemangioblasts differentiate simultaneously into 2 populations. The outer cells flatten into early endothelial cells, forming the walls of primitive vascular channels, the endothelial bags. The inner cells differentiate into the first red blood cells, filling those channels with hemoglobin. These are the blood islands. And when they connect and the primitive heart begins to beat, blood flows through a loose meshwork of these interconnected islands. Now here is the critical hemodynamic principle. Where blood flows fastest, the shear stress on the endothelial wall is highest. And that mechanical force is precisely what signals certain channels to mature and stabilize as major arteries and veins, while lower flow channels regress. The entire architecture of your vascular tree was literally sculpted by flow.
Here is a high-yield fact sequence that appears repeatedly on licensing exams, so memorize this migration pattern cold. Hematopoiesis, the production of blood cells, does not occur in one fixed location throughout development. It migrates through 5 distinct anatomical zones before permanently settling. It begins in the yolk sac and placenta during early embryonic life, producing primitive nucleated erythrocytes. It then transitions to the AGM region, the aorta, gonad, mesonephros, which seeds the definitive hematopoietic stem cell lineage. From there, the liver takes over as the dominant hematopoietic organ through mid-fetal life, enlarging massively to meet demand. This is why hepatomegaly is normal in the fetus. The spleen contributes briefly, then ceases. Finally, at birth, bone marrow assumes permanent, exclusive control. The clinical correlate? In conditions like thalassemia major or myelofibrosis, the body reverts to extramedullary hematopoiesis, reactivating the liver and spleen, a direct echo of this fetal migration pattern.
Now here is the molecular engineering rationale behind why hematopoiesis migrates. because each site produces a structurally different hemoglobin molecule, precisely calibrated for the oxygen environment of that developmental stage. Embryonic hemoglobin, produced by the yolk sac and AGM, has high oxygen affinity, necessary for capturing oxygen in the earliest low-supply environment. Fetal hemoglobin, HbF, produced by the liver and spleen, has very high oxygen affinity, specifically engineered to extract oxygen from maternal HbA across the placental interface. This is the Bohr effect in reverse. HbF sits to the left of the oxygen dissociation curve relative to HbA, binding oxygen more avidly at any given partial pressure. Adult hemoglobin, HbA, produced by bone marrow, returns to baseline affinity, optimized for efficient oxygen delivery to active tissues. The clinical bombshell, HbF persists up to 6 months post-birth, which is why sickle cell disease and beta thalassemia major don't present clinically at birth. HbF masks them completely until its replacement is complete.
Now we build the heart itself. At approximately day 18 to 19 of development, a specialized region of lateral plate mesoderm in the cranial portion of the embryonic disc differentiates into cardiogenic mesoderm, and its shape is unmistakable. A horseshoe, curving across the front of the embryonic disc with 2 arms extending laterally. When the embryo undergoes its critical craniocaudal and lateral folding, driven by the rapid growth of the neural tube and somites, these 2 lateral arms of cardiogenic mesoderm are swept toward the midline and fuse, creating a single midline structure. This fusion is what generates the primitive heart tube. At this earliest stage, the architecture is elegantly simple. Inflow at the bottom from the atrial primordia, a primitive ventricle in the middle, and the outflow tract, the truncus arteriosus, at the top, with blood flowing in a single direction, from inflow to outflow. Failure of midline fusion produces ectopia cordis, a heart positioned outside the thorax, one of the most dramatic congenital malformations visible at birth.
Here is a fact that should fundamentally change how you think about the heart. The heartbeat is not driven by the nervous system. It is an intrinsic autonomous property of the cardiomyocyte itself. This was demonstrated powerfully in cell culture experiments. Individual cells isolated from the cardiac mesoderm, placed in a dish with no neural connections whatsoever, spontaneously begin to contract. Each cell beats at its own rate, in its own rhythm. But the moment these cells make contact with each other through gap junctions, specifically connexin 43 channels, something extraordinary happens. They entrain. The fastest beating cell dominates and synchronizes all neighboring cells to its rhythm, creating a unified, coordinated contractile sheet. This is the biological origin of the sinoatrial node's pacemaker dominance. The SA node fires fastest, so it sets the rhythm for the entire heart. Clinically, this explains why ectopic foci in ventricular myocardium, if they fire fast enough, can hijack cardiac rhythm entirely, producing ventricular tachycardia by exactly the same entrainment principle.
Let's establish the exact anatomy of the primitive heart tube before looping begins, because understanding what folds is essential for understanding where each adult structure ends up. The tube has 3 distinct regions stacked in sequence from bottom to top. At the base is the atrial region. This is the inflow end, receiving blood from the developing sinus venosus and ultimately from the placental circulation. In the middle is the large primitive ventricle, also called the bulbus cordis, the primary contractile chamber at this stage. At the top is the truncus arteriosus, the outflow tract, which will eventually split to form the ascending aorta and the pulmonary trunk. Blood flows in a single direction, atrium to ventricle to truncus. The spatial challenge is profound. In the adult heart, the atria sit behind and above the ventricles. The aorta arises from the left ventricle anteriorly, and the pulmonary trunk spirals around it. None of this spatial arrangement exists yet. To get there, this straight tube must perform an astonishing 3-dimensional origami, cardiac looping.
Cardiac looping is complete by approximately day 28 of development, at a point when most women don't yet know they are pregnant. And yet by this moment, the heart has already accomplished one of the most geometrically complex transformations in biology. The process unfolds in 3 phases. Phase 1, lateral bulging. The straight tube first begins to bend sideways, bulging toward the right. This rightward looping, called dextral or D-looping, is genetically programmed by left-right asymmetry genes, particularly nodal and lefty signaling. Reversal of this direction produces situs inversus, or the cardiac defects of Cardigan syndrome. Phase 2, ventricular shift. The large primitive ventricle balloons forward and outward, positioning itself anteriorly. Phase 3, completion by day 28. The ventricle now sits anteriorly and inferiorly. The atrial regions shift posteriorly and superiorly, matching adult anatomy, and the truncus arteriosus comes to rest in front. The entire adult cardiac silhouette has now been established from a single straight tube.
Cardiac looping has given us the correct 3-dimensional shape, but the heart is still functionally a 2-chambered system, 1 atrial chamber and 1 ventricular chamber, with completely mixed oxygenated and deoxygenated blood. To reach the 4-chambered adult configuration, the heart must build complex internal walls, septa, that simultaneously divide the atrium into right and left, divide the ventricle into right and left, and split the truncus arteriosus into the aorta and pulmonary trunk. And here is what makes septation biologically astonishing. All of this construction must happen while the heart continues beating and pumping blood to a growing embryo that will die within minutes if circulation stops. There is no shutdown, no pause, no opportunity for internal scaffolding. The heart must renovate itself from the inside out, in real time, under full working load. When this process fails, even partially, the result is the congenital heart defects that affect approximately 1% of all live births. VSDs, ASDs, tetralogy of Fallot, transposition of the great arteries.
Atrial septation begins with the growth of the septum primum, the first wall. This thin, crescent-shaped membrane grows downward from the roof of the common atrium toward the atrioventricular junction. As it grows downward, it leaves a gap at its lower edge, the foramen primum. This gap is not a mistake. It is a deliberately engineered opening that maintains right-to-left blood flow during construction. Fetal blood must bypass the uninflated lungs, so atrial communication must be preserved at all times. As the septum primum continues growing downward and the foramen primum begins to close, sealed by tissue from the atrioventricular cushions, the embryo simultaneously initiates the next phase of its pressure maintenance strategy. Failure of foramen primum closure produces a primum-type ASD, the most common form of ASD associated with Down syndrome and atrioventricular septal defects. Recognizing this embryological origin is exactly why Down syndrome patients are screened for cardiac defects from birth.
Just before the foramen primum closes completely at the bottom, a new series of perforations appear in the upper portion of the septum primum itself. These small holes coalesce, merging together, to form a single new opening called the foramen secundum, positioned in the upper part of the first wall. The result is a seamless handover. The bottom gap closes, the top gap opens, and right-to-left blood flow is maintained throughout, without a single drop in pressure or a single interruption of circulation. This engineering relay, closing one hole while simultaneously opening another, is one of the most elegant solutions in all of developmental biology. It ensures the fetal circulation requirement, bypassing the lungs, is never compromised, even for a millisecond during the construction of the atrial wall. Clinically, if the foramen secundum forms too large and the subsequent septum secundum fails to cover it adequately, the result is a secundum-type ASD, the most common form of atrial septal defect, accounting for approximately 75% of all ASD cases.
With the foramen secundum maintaining blood flow through the upper portion of the septum primum,
The heart now builds its second reinforcing wall. The septum secundum grows downward from the roof of the right atrium to the right side of the septum primum as a thicker, more muscular, crescent-shaped structure. Critically, the septum secundum is intentionally incomplete. It never reaches the floor of the atrium. Its lower free edge stops partway down, leaving a permanent gap, the foramen ovale, positioned at its inferior border. The spatial arrangement is now a two-layer baffle system. The thin septum primum on the left, the thick septum secundum on the right with staggered openings. Blood can pass from right to left by squeezing through the foramen secundum opening, curling under the lower edge of the septum secundum. This one-way valve arrangement is functioning beautifully in every fetus right now, and it will seal permanently only at the moment of birth.
Let's consolidate the complete atrial septation terminology because exam questions test this vocabulary with precision. The septum primum is the first thin dividing wall growing down from the atrial roof. The foramen primum is the original temporary lower gap in the septum primum. It closes as the septum fuses with the AV cushions. The foramen secundum is the new upper gap that perforates through the septum primum just before the foramen primum closes. The septum secundum is the second, thicker, incomplete wall that grows from the right side, covering the foramen secundum but leaving an inferior gap. Together, the foramen secundum in the septum primum and the inferior gap of the septum secundum create the foramen ovale, the fetal cardiac bypass that allows oxygenated blood arriving from the placenta via the inferior vena cava to cross directly from the right atrium to the left atrium, bypassing the non-functional fetal lungs entirely. In 25% of adults, this foramen never permanently seals, producing a patent foramen ovale, a significant risk factor for paradoxical embolism and cryptogenic stroke.
And now, the most dramatic moment in cardiovascular development, the transformation that happens in the first breath. Throughout fetal life, left atrial pressure is low. The lungs are collapsed and receive minimal blood flow, so the two septal flaps sit slightly apart, allowing right to left flow through the foramen ovale. At the precise moment of birth, the newborn's first cry inflates the lungs. Pulmonary vascular resistance drops instantly and massively. Blood floods the pulmonary circulation for the first time. Pulmonary venous return to the left atrium surges, and left atrial pressure spikes dramatically above right atrial pressure. This pressure reversal physically forces the thin, flexible septum primum hard against the rigid septum secundum, like a door slamming shut from the pressure of wind. The two flaps are pressed together, instantly cutting off right to left flow. Over the following weeks to months, fibrous tissue grows and permanently fuses the two layers, forming the adult fossa ovalis. The fetal bypass has closed. The independent four-chambered circulation has begun. The cardiovascular system is now complete.